Welcome back, guys. We're going to cover section 4 today out of chapter 37. It's going to basically be some additional information on the adaptive immune system. Okay, so here are the 3 questions we want to think about as we're going through this. How do lymphocytes recognize foreign cells and molecules? How do lymphocytes produce specific responses to so many different types of cells and molecules? And how do they avoid mistaking the body's own cells and molecules for invaders? Okay, so one of the goals of the adaptive immune response is to recognize the complex molecules that the invaders can bring. So large complex molecules, so things like proteins, polysaccharides, glycoproteins that can trigger an adaptive immune response are called antigens. Okay, that's short for antibody-generating molecules, so it's going to make antibodies. Note here, antigens are not the living cells themselves, right? Those are what we call pathogens or bacteria or microbes. Those are the actual whole thing itself. The antigen is just the chemical, right? The molecule, okay? These are often located on the surfaces of invading bacteria, fungi, or other microbes. Over here, I believe this is a picture of an HIV virus. And you can see on the outside of it, there are some markers that are going to indicate that this is what this is exactly, right? It's got some proteins along the outside. There's some antigens, okay? So it's often located on the surfaces of these things, right? As you can see right there. Viral antigens may become incorporated into the plasma membranes of the body cells that they infect. So, and HIV is really, I guess if you want to say good or bad at this, but it's going to incorporate some of these proteins here into the cells in your immune system. HIV is a really horrible virus to get, which we'll talk about later on. But it basically just totally disrupts your immune system, and you'll see how it works. Anyways, dendritic cells and macrophages that engulf viruses or bacteria also display antigens from the microbes on their plasma membranes. So as these things begin to destroy stuff, they may incorporate some of these antigens on their plasma membranes. As it says right here, antigens aren't necessarily just coming from invading species or toxins. Any large organic molecule, including those from your own cells, may be an antigen and provoke an immune response. And sometimes this isn't the best thing. Autoimmune diseases, this is how they happen. Basically what ends up happening is the immune system cells begin attacking your healthy cells. So it gets tricked. It starts thinking that things inside of your body are antigens from a foreign body, and it's going to start attacking them. So how do you treat that? Well, you treat it by using drugs that turn off parts of your immune system or the entire immune system. So you shut off an immune system to make sure that your body doesn't attack itself. Okay. Another example here would be organ donation. So remember what MHCs are, major histocompatibility complexes on donated organs. Even on very close matches. So if you're not familiar with organ donation, it takes a while to get the right organ because you have to wait for somebody with very, very similar MHCs so that your body doesn't reject an organ. And even then, it can still mess up, can cause an immune response and a rejection of an organ. So if it doesn't match up perfectly, that person who's getting the organ transplanted into them might reject it, and their immune system might start attacking it because it doesn't look like it belongs. So immune system can attack donated organs because they seem foreign. So how do you treat this? Anyone who gets an organ donation will be treated with these immunosuppressant drugs. Basically, they're treated by drugs that turn off, just like autoimmune diseases, that turn off parts of your immune system or the whole immune system. So you shut off the immune system for a while, or indefinitely if it's really bad, so that that person who's getting the transplant doesn't reject that organ. So one of the things we've been talking about, if you've been paying attention to the news with COVID-19, I'm going to try to relate this as often as I can to what you guys are hearing about. People who are more susceptible to getting it, well, the elderly, people who have poor lung function, high blood pressure, people who are either in cancer treatment or have had cancer before are suffering more from this. And these two groups of people also have this problem because if you're using drugs to turn off the immune system, well, if you get sick with something like this, okay, these drugs have turned your immune system off, so you don't have as good of a way of fighting it. So if autoimmune disease people and organ donation people fall in that category of more susceptible to viruses, bacteria, things like that. So there's a kind of an important reason to bring that up. So just so you know some other things going into what's making COVID-19 pretty problematic. We'll get back to these later on in this chapter when we actually talk about autoimmune diseases, but if you're interested in what some of those are, here's a wide variety of them. So we'll come back to them, but I like to include it just so you can see what some of these are. And a lot of these you'll see commercials about. I apologize, my dog's barking outside. A lot of these you'll see commercials on when drugs are trying to treat this now. It's a very complicated system to try to treat these diseases. Okay, so let's say some sort of antigen gets into your body. What happens? Well, antibodies and T-cell receptors trigger your immune response. Okay, we talked about antibodies. We haven't really mentioned T-cell receptors yet, but that's what we're going to do here in the next few slides. So lymphocytes, your white blood cells, produce two types of proteins that bind to specific antigens and trigger an immune response, antibodies and T-cell receptors. Okay, I briefly mentioned this before, but we'll just quickly go over it again. Antibodies, these are produced by the B cells, right? These are your cells that are developed in the bone marrow. They're Y-shaped, right? They're composed of two pairs of peptide chains, one pair of identical large heavy chains, and one pair of identical small light chains. You can see what that picture looks like over here, okay? Last time I told you they were Y-shaped. Here's what it looks like very specifically. The heavy and light chains consist of a variable region and a constant region. And I did mention this before, but just so you can see it, the variable region is the tips of this Y shape. Okay, these bits right there, right here, and right here, those can change from antibody to antibody. The rest of this, okay, the rest of this piece is all the same. So why is that important? Well, this will change. The ends of these antibodies change to recognize a specific antigen that's going to fit exactly in this slot. Okay, here, or whatever this thing is, okay, this is what's going to bind to it. This is a very, very specific shape in here, okay? That's why it recognizes a specific pathogen, okay? So as we were saying a second ago, the heavy and light chains combine to form the arms and the stem. The tips of the arms are the variable regions, right there, right there. Okay, each binding site has a particular size, shape, and electrical charge, which allows only certain antigen molecules to fit in and bind. So it's going to look for a very specific antigen. Every B cell produces its own unique antibodies with variable regions different from other B cells. So they're going to target very, very specific antigens. Each one makes their own special type of antibody. Okay, so if the variable ends are what recognize the antigen, what's the rest of this thing do? The stem of it tells the antibody where it's going to be in the body, like what role it's going to play in the immune response. So where it is and what its specific job is. There are 5 main classes of antibodies, IgM, IgD, IgG, IgA, and IgE. Okay, you don't have to memorize all 5 of those. The two kind of ones that come up, that will probably come up again for us, IgG types of antibodies are the antibodies that fend off pathogens, okay? IgE, those are the ones that are responsible for allergic reactions. So if you have like a peanut allergy, a nut allergy, shellfish allergy, what that means is that you have antibodies, okay, IgE antibodies, that go into full immune response when you consume or touch or whatever it is, however you are allergic to that particular product. Okay, so that's what those are. All right. So the function of them, we've mentioned this before, but just again, recognize foreign antigens and triggering the response against invaders, helping to destroy the invading cells or molecules that bear the antigens. This is just a picture out of your textbook, so here's kind of just what a B cell might look like. Here the antibodies are on the outside of a B cell. Again, they're very, very specific for a particular microbe or pathogen. So here's your microbe. The antigen is this bit here along the outside, and so your antibody is going to recognize a very specific antigen and bind to it. That's going to tell this B cell that, hey, we've attached to something that doesn't belong here, okay, and that's going to trigger responses. Okay, so enough about antibodies. What about T cells and T cell receptors? So they have there's a different type of protein called T cell receptors that recognizes and binds to antigens. This is, by the way, a picture of a T cell over here. Every T cell produces these receptors, and they're different from those of other T cells. So just like how the B cells produce antibodies, T cells produce T cell receptors. Okay, so just trying to help you keep things clear, right? B cells produce antibodies, right? T cells, hopefully this one's easy, right, produce T cell receptors, okay? Trying to keep everything as easy as we can here. So when an antigen binds to a T cell receptor, the T cell becomes activated in ways that depend on the type of T cell. So these bits here around the outside of the T cell, these receptors, depending on what hits it, is going to activate this T cell to do something. So the adaptive immune response can recognize millions of different antigens. Okay, so throughout your lifetime, you're going to be challenged by many potential invaders. You're going to come in contact with a lot of stuff. Someone's going to cough on you. You eat contaminated or moldy food. You have a disease-carrying mosquito, something like that. All right, West Nile, right, an example here. You're just going to come in contact with a lot of stuff. So in addition to the pathogens you've experienced, B and T cells randomly synthesize millions of different antibodies and T cell receptors. Okay, so when you come in contact with these pathogens and antigens, your body responds by creating antibodies or T cell receptors to remember these things, right? Okay, but in addition to the ones that they've already encountered, they are just randomly synthesizing antibodies in hopes of getting one or two of them right. Okay, so these are produced randomly. So you might just by chance have the correct antibody or T cell receptor without ever having encountered a particular disease. So you might hear about, well, why are some people not getting this, symptoms of COVID-19, or like why do some people not have as bad of symptoms, or why is their actual experience of the virus not as bad? Well, perhaps, okay, they just by chance had B and T cells producing the right antibodies and T cell receptors just randomly. Okay, that's certainly a possibility there. Okay, so there's estimated that there might be 100 million different antibodies and T cell receptors in the body. Okay, that's not to say that you've come into a contact with 100 million different pathogens. You probably come in contact with a few million different ones, maybe more. But you also have these random ones that are getting made just in case something comes in contact that you've never met before. Maybe there's a chance you guessed right. Okay, so because of this, most invading antigens have a high probability of encountering an antibody or T cell receptor that will match close enough to mark it as foreign. Okay, these randomly created antibodies and T cell receptors might be close enough that it can still get marked as foreign. So I hope that makes sense. You have, B cells that we said make antibodies. Okay, you got T cells, which makes T cell receptors. Those often come in the response after getting a particular virus or bacteria or fungal infection. And then you have these randomly generated ones also by B and T cells, just hoping to get it right. Okay, and then part of those questions that we asked at the very beginning, why don't your own body's antigens trigger your own immune response? So the continuous presence of the body's own antigens during immune cell development is part of that answer. And modulation of the immune response by regulatory T cells. Okay, that gets into some fancy wording. We'll see what that is here in a few minutes. But this first point right here, as your B cells and T cells are developing, okay, when they first form in the bone marrow, remember they all start in the bone marrow, B cells finish there, T cells go to the thymus, they can bind to antigens, all right, as they're forming, but they're not yet able to trigger the immune response. So they can bind to antigens, but not do anything. If these immature cells bind to an antigen, they undergo, if they bind to a self antigen, something that recognizes it as belonging to you, they undergo apoptosis, which is programmed cell death. They basically kill themselves off because they would start producing antibodies to your own antigens, which would not be good. Okay, so ones that bind to self-antigens, things within you, kill themselves off. Okay, immature B and T cells are constantly exposed to the body's own antigens, so the ones that produce self-reactive antibodies or T cell receptors are killed off before fully developing, which is what we were just saying right there, that if they are the ones that accidentally get stuck with these self-antigens sticking to them, they die off. Okay, we only want the ones that are going to react to foreign bodies. So ideally, only B and T cells that did not react to the body's antigens survive. Okay, they mature. However, in reality, sometimes some of them skip through. Some of them might survive. So you might end up accidentally with some B and T cells that still attack your own cells in your body, right? You kind of get this autoimmune disease problem is what this can lead to. It's okay. The body does have a way to try to stop this. There's a type of T cell called a regulatory T cell, and their job is to prevent these cells from remaining self-reactive lymphocytes from attacking the body's own cells. So anything that gets left over, they're trying to turn them off. Okay, these regulatory T cells are trying to turn off the bad B and T cells, the ones that are accidentally binding to your own antigens. And that's it for today.